Hello, my name is Хайбулаева Амина. I'm a first-year student of Saratov State Medical University. I study the faculty of pediatrics. My report focuses on one of the most controversial topics in medicine nowadays, gene editing and how it can shape our future. Gene editing is a technology that can change DNA sequences at one or more points in the strand. Scientists can remove or change a single base or insert a new gene altogether. Gene editing can literally rewrite DNA. The idea of using gene editing to treat disease dates to at least the 1950s and the discovery of the double helix structure of DNA. In the mid-20th century era of genetic discovery, researchers released that the sequence of bases in DNA is passed faithfully from parents to offspring, and that small changes in the sequence can mean the difference between health and disease. Since then, various methods of gene editing have been created, the most popular of which is CRISPR-Cas9, discovered in 2012 by American scientists. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats and was first discovered in bacteria like a part of bacterial immune system defending against invading viruses. The system consists of two key parts: a CRISPR-associated Cas nuclease, which binds and cuts DNA, and a guide RNA sequences, gRNA, which directs the Cas nuclease to its target. Imagine that Cas is a pair of scissors and gRNA is a hand that controls them. It works as follows. When a cell is attacked by a virus, a fragment of viral DNA appears in it. To remove it, the cell uses CRISPR-Cas9, which cuts out the viral DNA. This fragment is stored in the bacterial genome along with the fragments of other viruses that previously infected the cell, immune memory. If reinfected with the same virus, the bacterium can recognize and destroy it using CRISPR. CRISPR-Cas9 gene editing works by creating double-stranded breaks in the DNA. The lost DNA sections of the cell can be repaired, but the viral DNA doesn't have the ability to recover. In addition, it is possible to add a DNA template to the mechanism of CRISPR-Cas9 and correct the cellular gene. Korean scientists have introduced mutations in the DNA of active neurons in the brains of mice suffering from Alzheimer's disease, which reduced the symptoms of the disease in adults. To do this, mice were injected into the brain nanocomplexes with the CRISPR-Cas9 protein, which was configured to cut out the gene that provokes the development of this disease. And in the Novosibirsk Laboratory of Epigenetics, with the help of CRISPR, work is underway to obtain isogenic stem cell lines that simulate Huntington's disease. Huntington's disease is a congenital progressive neurodegenerative disease, the clinical manifestation of which is involuntary movements of the arms, legs, trunk, often facial muscles, which can be by mood and behavior disorders, as well as memory and thinking. Gene editing is still a controversial topic. Unless governments work together with scientists to regulate its use, it could become another technology that benefits only the wealthiest people. Secondly, it comes with risk. The first case of illegal implantation of genetically edited embryo was reported in 2019 in China and led to the imprisonment of 3 scientists. The scientists had attempted to protect twin fetuses from HIV being passed on by their father. But when other scientists read passages from an unpublished paper written by the experiment led about the twins, they feared that instead of introducing immunity, the researchers probably created a mutation whose consequences are still unknown. In my opinion, there is no doubt that CRISPR is the key to the medicine of the future, and with its help, we will be able to cure disease that now seems to us incurable. But laws and regulations surrounding that technology have to be strict. Scientists need to work closely with laws and policy makers to ensure that technology can be used for the benefit of mankind while minimizing the risk. Thanks for your attention.